Hello everyone, my name's Gerry Davies. I'm a Professor of Infection Pharmacology here in Liverpool in the UK, and I'm very grateful to ESM for the opportunity to speak to you this World TB Day on the subject of the use of time to positivity in automated liquid culture systems as endpoints in TB clinical trials. So first of all, I'm going to say a little about the way we use microbiological endpoints in TB trials and some of the issues and choices there. I'll touch on some important, I think, basics of quantitative mycobacteriology methods in the laboratory, which are relevant. Then we'll talk about the opportunities and the challenges and characteristics of automated liquid culture systems, which are also important to understand. Then we'll review some of the clinical studies that have been done comparing serial sputum colony counting on solid media with time to positivity in liquid culture. We'll talk then about how we handle that data statistically and some of the issues there. And finally, I'll say a little about some recent mechanistic insights which I think are important in understanding what we do with this data and the inferences that we can make. So to start at the very beginning, for this audience, it doesn't need stressing that TB is a chronic bacterial infection which exhibits persistence phenomena. In other words, despite participants in clinical trials and patients in programs having very favorable microbiological data, culture converting being negative on mycobacterial culture at the end of treatment, it's well known that if treatment is too short or ineffective, that people will relapse either sooner or later, and that because of the lack of durable host immunity, they may also be reinfected. Underlying those simple concepts, of course, is not just a qualitative, but an underlying quantitative paradigm, which is similar to the kind of viral dynamics models that we use in HIV and in hepatitis C. And some years ago, I reformulated some of the endpoints that are quite commonly used in phase 2 and phase 3 clinical trials in just such a framework that you can see here. Importantly, emphasizing the limit of detection of the bacteriological methods that we commonly use. Underlying these ideas are some concepts of the way in which anti-TB chemotherapy works, going right back to the 1970s. And these were jointly developed by Denny Mitchison, a microbiologist, and by George Canetti, who was a pathologist. The chronicity of TB, particularly pulmonary TB, of course, leads to very varied microenvironments within the lung, in the kind of necrotic and hypoxic areas inside TB lesions. But that also leads to heterogeneity within the bacterial population, the majority of which is probably still able to grow in a continuous fashion in a logarithmic phase, but significant subpopulations of which probably are not growing at all, or growing at a very slow rate, or perhaps only grow intermittently during treatment. And so the theory goes that a combination of drugs whose pharmacodynamic activity is focused on these different subpopulations is what's important in successful multi-drug chemotherapy. Some years ago, we reformulated the subpopulations idea in a more operational way. Because during treatment, of course, the first thing that happens is that people become negative bacteriologically on solid media, followed by culture conversion in liquid media, and then organisms are often only able to be recovered by addition of resuscitation promoting factors to any kind of media. And as time on therapy proceeds, those populations become smaller and smaller until they are ultimately eliminated by drugs and or the host, resulting in a stable cure. So the idea of operationalizing that subpopulation concept leads us to think that perhaps there's something about liquid culture which is telling us about populations of bacteria that don't grow on solid media that might be important for establishing that somebody and predicting that somebody is going to experience a long-term stable cure. And when we talk about quantitative microbacteriology, there are a few things I think to say just as a preamble, which may not be immediately obvious, but which can really influence the data and also the way that the data are analysed. This is some data we collected some years back in Malawi, looking at the volumes of sputum that people produce when they're asked to participate in clinical studies. And you can see here that the mean volume of sputum is of the order of 10 mils, 10 or 12 mils at baseline before people have started treatment, and that in most people, production of sputum persists even as late as 2 to 3 months. But the maximum and minimum volumes of sputum vary quite a bit, and in particular the maximum decreases from about 80 mils to about half that after 6 to 8 weeks of therapy. Also important to note that many people stop producing sputum quite early on. So after 2 or 3 weeks, probably more than half of people are not producing any sputum at all, and we're not able to follow numbers of organisms in those patients thereafter. So that the volume of sputum in individual participants can vary from as little as 2 to as much as 80 mils is important, because there can be a log difference if we use concentration methods. There can be a log difference in the number of organisms just based on the volume of sputum alone. And the different kinds of quantitative microbacteriological methods that we use are somewhat different from those we normally apply for diagnostic purposes. So the usual procedure, of course, is that when the specimen is concentrated in the lab and then decontaminated with NaOC sodium hydroxide, that often the entire sample may be pelleted and then centrifuged and spun down, and that a small amount, usually about 0.1 mils, will be inoculated onto a slope or into a liquid culture bottle. And so those dilution factors are quite important in determining whether samples are comparable from person to person and from occasion to occasion over time. For the purposes of phase 2 trials in TB, the classical method is to homogenize the sputum, usually with dithiotretol, and to take a single mil, which is then processed and plated out onto selective 7H11 plates, which contain the classical antibiotic mixture of polymyxin, amphotericin, ticagrelin, and trimethoprim. And that helps to prevent contamination that would otherwise be prevented by malachite green. And the same is true, of course, in liquid culture systems. So when we compare colony-forming units derived from serial sputum colony counting, these methods are precise and standardized. We also need to apply the same thinking to liquid culture and make sure that we are processing the samples and the volumes of samples in the same way, so that the results will be, broadly speaking, comparable. That also relates to the limit of detection of the two methods, of course, depending on the size of the inoculum. And in fact, depending on how you inoculate the liquid culture bottles versus how you prepare the inoculum for the plates, in fact, the limit of detection of serial sputum colony counting could be a bit lower than it would be for automated liquid culture, depending on the protocol. So some of those details are important, and for individual clinical trials, we need to look at those details carefully in the methods to make sure that we understand whether they are in fact comparable. So the first stage of drug development in which serial sputum colony counting and quantitative microbacteriology became prominent was in the first studies to be conducted in humans, phase 2A, and that resulted in the concept of early bactericidal activity, which has long been in use and is frequently misunderstood and can be misleading. But as it was originally used, EBA was essentially the fall in colony-forming units over the first 2 days of therapy, using quantitative methods on solid media. And using that approach, it was possible to dose titrate monotherapy for individual drugs in quite a precise way. And you can see here some of the results of the early studies on the same graph. And in particular, I draw your attention to isoniazid, the green line, which shows that it's possible to define both the minimum effective dose of isoniazid using this methodology and also the maximum dose. So once you get to about 300 milligrams daily on average, then there is a plateau in the activity which doesn't really increase much as the dose goes up. That's probably not true for various rifamycins, which you can see here, rifampicin, rifabutrin, and rifapentine, and also for quinolones and aminoglycosides, where there's no obvious maximum reached, at least at the doses that were studied in these studies. So early bactericidal activity established itself early in the drug development pathway and has been a useful tool for study of monotherapy of some drugs, although notoriously, of course, there are drugs which show little or no early bactericidal activity, among them PZA, clofazimine, and arguably bedaquiline. So the development of the methodology required that our focus be extended a little bit beyond just the first 2 days, and perhaps needed to take a look at combination drug regimens. And 15 or 20 years ago, phase 2b studies, the next chain in drug development in TB, the next link in the chain, was traditionally carried out using 2-month culture conversion with pretty large studies, usually of the order of 120 to 150 patients per arm. We helped to develop a method which extended the concept of EBA to longer studies using combination therapy, and showed that in many of those studies there was quite a characteristic biphasic response, where the rate of elimination of organisms slowed down after the first week of therapy, consistent with the concept of the presence of persister organisms, which were more tolerant perhaps of antibiotic action and were eliminated more slowly. And that seemed to be true both in African studies and in some Asian studies. And that methodology has gradually become more widely established in the field and is now typically used to select amongst combination regimens for prioritization in phase 3. The first such study really to show that was the Oflatub phase 2b trial, in which we first applied this kind of approach, and the precision was such that we were able to demonstrate that actually both gatifloxacin and moxifloxacin, when substituted for ethambutol in the standard first-line regimen, accelerated culture conversion by about 25%. And in fact, both of those regimens went forward into phase 3 trials, and despite an apparent initial disappointment when the results of the individual trials were announced, it transpired that, in fact, in the most favourable prognostic subgroup in those trials, about 2/3 of those patients, in fact, that regimen was capable of shortening treatment to only 4 months. There are some of the issues with this data which are important to mention, of course, are this concept of the limit of detection. This original analysis was not completely robust to the presence of missing data and handling the limit of detection explicitly. And so we subsequently looked at the impact of accounting for the presence of observations below the limit of detection, which can be calculated as 10 organisms per mil according to the dilution factors, and using statistical methods which account for the distribution of observations below the limit of detection. We can see that those results are pretty robust. In fact, the differences between regimens are pretty robust to accounting for the limitations of the underlying quantitative mycobacterial methodology. We've also subsequently shown that these methods are precise enough also to distinguish between doses of individual drugs in the same way, in fact, as EBA studies can. And these are the results published some time ago now, of the high rift study in Peru. And you can see here that we were able to detect changes in the dose of rifampicin in the first-line regimen from 10, 15 to 20 milligrams per kilogram using this approach. And crucially also to show that there was an increase in activity with rifampicin pharmacokinetic exposure. So there are many other studies that I could mention conducted by other groups, other developers, but the quantitative approach to doing phase 2A and phase 2B studies is now something that I think is standard in the field. And I think it's fair to say that over the last decade, though the laboratory methods have remained much the same, the statistical approach to analysing that data and designing these trials has improved immensely and has led, I think, to refinement of the methodology in drug development. But it's often said that because serial colony counting is a very specialist technique and methodology and can really only be carried out to a high standard in labs that have experienced staff and good facilities. And so there really are, you can count on the fingers of one hand, the laboratories which can support EBA studies in TB drug development. And so the discussion has been around whether automated liquid culture systems, and in particular midget, could substitute for this rather laborious and labour-intensive colony counting methods on solid media. Why do we think that? Well, in general, people feel that liquid culture tends to have a higher sensitivity and a lower limit of detection, although, as I mentioned, that depends on how you process the sample, of course. But I think everyone knows that it's a better medium. Broth culture will often be a better medium for recovering damaged or fastidious strains, or those that have been recovered during therapy. It's attractive that automated liquid culture systems have perhaps a more standardized sample processing protocol and the reagents that go into the preparation of the media, and having a machine which automatically monitors the progress of the culture is something which is inherently very attractive, but importantly also provides quantitative data without the investigators having to do any other work. Perhaps the only downside to adopting liquid culture would be its probably greater vulnerability to contamination, despite the use of a similar antibiotic mixture that we use in selective colony to make the selective 7H11 media for colony counting. So what do we know about liquid culture? Some years ago I did quite a lot of experiments using another automated liquid culture system, the MB-BAC-T system by Organon Technica, but I think this is generally true for most liquid culture systems, that if you look at the range of linearity of days to positivity and you calculate the numbers of organisms that you're putting into those systems. So these are not clinical samples. These are simply a broth stock of the reference strain H37RV. You can see that there is a defined range of linearity that is from about approximately 1 million organisms down to slightly less than 1, and that outside those limits what you see is a deviation from linearity in the time to positivity. At the bottom end of the range there would seem to be a lower limit, perhaps represented by the minimum generation time of the particular strain, and at the top of the range you see an increase in the time to positivity, perhaps related to stochastic effects or Poisson effects as to whether an organism is actually inoculated into the bottle at all, or a lack of cooperativity amongst strains as they begin to exit lag phase. But we'll talk about that in a moment. So there are potentially some quantitative limitations, as there are with colony counting on solid media, depending on how the dilution scheme is arranged. There are also some differences between strains. So this is just a simple example where we did a similar calibration curve in MB-BAC-T for H37RV and the avirulent strain H37RA, and you can see that there are some differences both in time to positivity at the same inoculum, but also that those differences diverge as the inoculum becomes smaller. So all strains are not alike when it comes to growing in liquid culture media, and indeed liquid culture media are the preferred environment for doing fitness competition experiments between different strains, and that's something we should be aware of. What do we know then about the use of colony counting as against automated culture in EBA studies? So this is data that published by the preeminent EBA centre in the world in Cape Town from Andreas Diacon's group. And you can see here a broad correlation between measurements that were taken from people participating in many different EBA studies on many different regimens, between solid quantitative mycobacterial culture and time to positivity in automated culture. And you can see that broadly speaking there is quite a good correlation if you simply try to predict using a log-log plot what how many CFU would be predicted from any given TTP measurement. You can see that broadly speaking the correlation is pretty good up to a point. So if you go above about log 6.5 organisms, you can see that it tends to be the case that the number of CFU predicted from the TTP measurement is perhaps a bit less, and that may be something to do with the technical characteristics of the system. You'll also note that there are a few measurements over here at the bottom end of the range which also seem to be somewhat off the line, related to the considerations that we discussed just now. But broadly speaking, these investigators were pleased to see that for a broad linear range there does seem to be quite a good correlation between time to positivity and CFU in these studies. And that's true if you look at the individual measurements within individuals. So for each individual, this is the EBA for the first week of therapy on different combination and monotherapy regimens, calculated from TTP or from CFU. And again, you can see a broadly linear correlation with a relatively high proportion of the variance explained. And that's also true when you look at different regimens. And so these are different test regimens in these EBA studies and how they correlate between TTP and CFU at a population level. And again, the linear correlation is promising. Can that be extended to longer studies, given what I said previously about the changes that we see during therapy in bacterial subpopulations or putative bacterial subpopulations? Well, one of the first data sets that I was lucky enough to come across was the Adjuvit study conducted by Adrian Martino in London, which was a trial of vitamin D supplementation during TB therapy. And this was one of the first data sets in which we were able to try to define a statistical model for TTP data. And you can see that in this case, the best fit model was in fact a linear one, unlike the biphasic ones that we saw for some of the serial sputum colony counting studies. And that may have something to do with the decontamination that was conducted in NHS diagnostic labs for the purposes of this study, potentially eliminating some of the logarithmically growing organisms before they were ever inoculated into the bottle. But the fit of this model is good. The trial itself failed to show that vitamin D accelerated culture conversion in liquid culture, but it did give us some insight into how to handle this sort of data. Similarly to growth on solid media, there is an issue, of course, of limited detection with the typical diagnostic limit of 42 days in the machine being applied in almost every clinical trial. What's quite interesting about that is that, well, there are two things. The first is that obviously to handle that we need to use similar but allied statistical methods to handle the limit of detection, because we are handling a limit of detection that is a number between 42 and infinity rather than a number that is between 10 and 0. But using those kinds of methods, you can show that actually the inferences derived from the trial are pretty robust to the limit of detection in the automated liquid culture system. But one thing that you also notice is that there are very few observations, in fact, between about 28 to 30 days and day 42. And there's that the distribution of, at each time point, the distribution of time to positivity measurements has a bit of a skew in this area, but that the data are rather sparse, which is probably worthy of further exploration. In other words, that the limit of incubation that we use for diagnosis may not be fully reflective of what we need to model data in clinical trials. The other question that often comes up about both serial sputum colony counting and also time to positive data is whether they are related to longer term outcomes, specifically to culture conversion at 2 or 6 months, or indeed to long-term stable cure. We conducted a couple of studies in Malawi to look at that question, and what you can see here are the results of the sputum study conducted by Derek Sloan, in which a cohort of people on first-line standard chemotherapy were followed according to their prognostic characteristics, and with some other biomarker studies that we'll talk about presently. And what you can see here on the left are the individual trajectories derived from the mixed effects linear model. And you can see the darker lines are people who had unfavorable outcomes, and the lighter ones are those who ultimately had a stable cure. And you can see, just looking at the plot, that perhaps there are some differences with slightly shallower trajectories for those who had an unfavorable outcome, and steeper for those who were cured. And indeed, if you look at those, if you look at the average values in those groups, you do find that there does seem to be a difference, with a steeper increase in time to positivity per week of therapy for those who were ultimately cured, as opposed to a more shallow one for those who weren't. And worth saying that this was a cohort, of course, that was selected to be sputum smear 2+ positive or more, and the overall relapse rate in this cohort was about 11% because of the distribution of unfavorable prognostic factors. These were people who were selected in a group which has a relatively high relapse rate. Those studies continued in the SPIT study conducted by Andrew McCallum, and similarly in that study we were able to show that there was a statistically significant difference in 8-week culture conversion between those who had an unfavorable outcome versus those who had a stable cure. But in this study it was not so clear, actually, that the long-term outcomes were different between those two groups. Andrew's focus was more on the use of TTP slopes to predict, to the use of TTP slopes to evaluate the effect of rifampicin and isoniazid pharmacokinetics, intrapulmonary pharmacokinetics, on outcomes. And in fact, he was able to show, using this methodology, that the higher your rifampicin and isoniazid pharmacokinetic exposure in epithelial lining fluid, the better your outcome. So TTP seems to have some promising data behind it, perhaps not as extensive yet, but a similar range of use cases that we've described for solid media quantitative colony counting. But there have always been some concerns and discussion about whether what we're observing in terms of the model that we're fitting to the data really reflects the underlying bacterial physiology. And Ruth Bonus, in an interesting study where she looked at the data from the Panacea HiRIF1 study, she looked simply at the relationship between the colony counts on solid media and the time to positivity in automated culture, which had been collected, both of those measurements been collected for all the patients and at all time points. And she simply plotted out the relationship between the two according to the number of days on treatment. This was a relatively short-term study, only 14 days, an EBA study for 14 days using monotherapy. And what she showed was that the slope of the line describing the relationship between CFU and TTP changes with treatment. And in fact, the difference for the same number of colony-forming units at day 0 as against day 14, there was a difference of almost 5 days in terms of the time to positivity in the liquid culture system, according to days on therapy. And not only that, the slope of the line at different times was seemed to be different from that at baseline, a little bit analogous to the kind of data that I showed you with different strains in liquid culture. That variation appears to be something which is induced by therapy. And Ruth then went on to try to amend the kind of prediction equation that I showed you previously by using different nonlinear functions. In fact, she ultimately chose to use a Gompertz growth function, which is a fairly flexible nonlinear function, and she came up with a prediction equation that looks like this, with several parameters, but which was more accurate in predicting the number of CFUs from TTP than a linear equation would have been, as you can see here. So there are ways of simply representing that change in the relationship between TTP and CFU over time by using a time-varying function that's non-linear in nature. An alternative approach has been to take a more mechanistic kind of view of it, or semi-mechanistic view of it, and here is quite a complicated model that was recently published, which tries to capture the relationship between CFU and TTP, and also to incorporate pharmacokinetic considerations as well. But in essence, the important thing to understand about this is that it's driven by the total bacterial census, that is to say the number of rapidly growing and slowly growing organisms in this putative bacterial growth model, and that is translated into a number which drives the time to positivity, and then that is linked to essentially a time to event model which predicts how long it will take for the system to trigger. Now, I have some concerns about this model. I think it probably is over parameterized, but I think the ambition of it is laudable to try to understand what the relationship between those organisms that grow on solid media and those organisms that grow in liquid media actually is, and whether we can capture that in a more accurate mathematical description. But however we represent the results of these data, whatever empirical functions we can fit to them that are simpler or more or less complicated, the question remains whether we really understand what's happening inside liquid culture systems. And a couple of pieces of work that we've done perhaps inform that to some extent. The first comes from the sputum study that I showed you, which was a biomarker study, really trying to understand whether we could directly visualize persister organisms, or at least organisms that have a persister phenotype, in clinical specimens during therapy. And Derek was able to show that with time, during the first month of therapy, the number of organisms that have triacyl glycerol droplets inside them and can be visualized by Nile red staining tends to increase, and so that those specimens tend to be enriched for those organisms. But more importantly than that, the people who ultimately experienced an unfavorable outcome failed to eliminate those organisms as quickly as those who had a stable cure. And so the outcome of that study really was that we were able to show that there are some differences in bacillary phenotype in patients during therapy, and we're not completely clear how that change may influence time to positivity. that may be one of the factors that's responsible for the changes that Ruth Bonus saw in the HIRIF1 dataset. Furthermore, in another study, the panbiome study that followed these, David Barr looked very carefully at what happens to colonies on solid selected medium plates and compared that to the time to positivity in paired samples in the midget system. And what was really interesting about that was that you did see a change in the rate of appearance of colonies and some differences in their appearance. And David, photographed plates quite intensively over the first 2 to 4 weeks of therapy, and was able to calculate the size of the colonies using photographic methods. And what he showed was that though there were not really strong correlations between the mean colony growth rate, or indeed even the mean colony lag time in terms of the time to positivity, what did seem to have quite a strong determination of time to positivity in midget was the time to appearance of the first colony on solid media. And so it may be that in terms of the model that I showed you with multiple bacterial states, that actually it's the presence in the sample of any logarithmically growing organisms that probably determines the time that it takes to trigger the automated liquid culture system. So quantitative microbacteriology is now, an important workhorse really for us in early phase clinical trials in TB, and it's difficult for us to imagine doing without it. We think we understand solid culture reasonably well, and though it looks as though time to positivity in liquid culture may be more reproducible and has a lot of attractive characteristics because it's widely available and more standardized, and though there is a broad correlation in measures of microbacterial load derived from the two, we know there are some limitations in limits of detection and the linear range in automated liquid culture systems, and we need to think about those carefully in terms of designing our endpoints. We also need to represent those data faithfully when we analyze them, and there are a number of proposals as to how those data would be most accurately modeled. And I think more work needs to be done on that to make sure that we are faithfully representing the data and getting the inferences out of them, and that the inferences that we are making from them are reliable. But I think we also need to have some deeper study of what it is that really determines time to positivity in liquid culture systems and how that varies, both between participants, between the strains they harbour, and over time during treatment. So I'll stop there and just acknowledge some of the people who carried out a lot of this work, particularly to Denny Mitchison and Christian Leonhardt, who got me involved in the Offlatub study, Carol Mitnick and Chuck Peliquin, who were my co-PIs in the HIRIF study, and David Coleman, who did a lot of the bacteriological work, and to Derek, Andrew, and David, who carried out a lot of the work that I showed you in the latter part of this talk. Thank you very much for your attention. If this interested you and you have any questions, please don't hesitate to contact me on the email at the bottom of the slide.